Hello friends, in this video we will discuss about the steps in microarray. So if you haven't watched the video on the introduction of microarray, the principle and the types, please do watch it before you go with the steps. First, we will discuss what is the requirements of a DNA microarray. A DNA chip, a target sample, and this sample is being fluorescently labeled. Then we need fluorescent dyes, probes, and a scanner. Next, we will discuss about the steps. Here we are going to discuss about the cDNA based microarray. So the first step in cDNA microarray is the collection of samples. A sample can be a cell or even a tissue of an organism. And two types of samples are being collected, that is a healthy cell and an infected cell. These two are collected in a purpose of comparison. So the first step of cDNA microarray is the collection of sample. So the next step is the isolation of mRNA. So the RNA is being extracted from the sample which we took, and this separation is done by a column or a solvent like phenolchlorophyll. So using this technique, a RNA is being extracted, and once a RNA is being extracted, it is separated into layers. That is, it is separated into mRNA, rRNA and tRNA. From this, we isolate and take the mRNA, leaving behind the rRNA and tRNA. So we know that the mRNA has a poly A tail, and since the mRNA has a poly A tail, column beads are used for the separation. That is, the beads with poly T tails are used to bind the mRNA. So the mRNA comes and binds to these beads which are poly T tails. So after the extraction is done, the column is being rinsed with buffer so that we get the mRNA. So the next step is the creation of the labeled cDNA. So the third step is creation of labeled cDNA. So in order to create a cDNA, the mRNA has to be reverse transcribed. So after a reverse transcription, the mRNA becomes the cDNA. So both the samples are incorporated with different fluorescent dyes. So we get different colored fluorescent cDNAs. So this is done in order to distinguish the sample category. That is, if the healthy cell is given a red color, the unhealthy cells are given green color. So in this way, we can distinguish the sample category of the cDNAs. The next step is hybridization. So this diagram shows how a cDNA is being created. That is, how a labeled cDNA is being created. From a mRNA, a cDNA is being created by a reverse transcriptase. In this step, a labeled poly T primer is being added. So, this primer which is added is labeled. So, the entire cDNA which is produced is being labeled. So, this is how from a mRNA, a cDNA is being produced by reverse transcriptase. So, in this step, the labeled cDNA, that is, the cDNAs from both the samples, are placed to a DNA microarray. So, here the cDNA gets hybridized to the complementary strand. So, the unbound sequences are removed by washing. The last step in DNA microarray is collection and analysis. So the collection of data is done by a microarray scanner. So this scanner consists of three parts: a laser, a computer, and a camera. So the laser excites fluorescence of the cDNA and it thus generates the signal. So when a laser scans the array, the camera records the images. So the computer then stores the data immediately when the results are being produced. So these data produced are then analyzed. So the difference in the intensity of the colors of each spot determines the character of gene in that particular spot, which means that it is the intensity of the color which determines the character of a gene. This diagram shows the overall steps in a DNA microarray. Here, two cells are being taken, that is, two samples, a normal cell and a cancer cell. These samples are cultured, and after they are cultured, RNA isolation is being done. That is, the mRNA is being obtained. After the mRNA is being obtained, they are done a reverse transcription. In this step, the cDNA is being produced, and the cDNA is being labeled, and this labeling is done by fluorescent dyes. Both the cells are given two colors, that is, for example, the normal cells are given a green fluorescence, whereas a cancer cell is given a red fluorescence. The two cDNA is being mixed and they are hybridized to a microarray. In this step, the hybridization occurs, that is, the cDNA comes and binds to the complementary strands. And after this step is done, the last step, which is the scanning and analyzing step, is done. The result of this step is that the green color shows that they are the normal cells, whereas the red color shows that there is disease cells only, whereas the yellow color shows the presence of both the cells, that is, the normal cell and the cancer cell, whereas the grey color shows that there is no presence of any of the cells. So, based on the intensity of the color, we can say the character of the gene. I hope the concept of microarray is clear. For more such videos, do subscribe to our channel. If you like the video, please like and share with your friends.